common genetic variants in cardiac arrhythmias. And you all are aware that at the initial phases, the genome-wide association studies, and many genomic studies were focusing on associations and very little on the causation. And we are just beginning to emerge, and I'm happy to share some examples where it is already possible to move from a pure association with common genetic variants to the causation. First, the disclosure is the various funding sources that support this work, and then I want to give credit to some of my colleagues that contribute to the work that I present here, primarily Moritz Sinner, Fritz Beckmann, Martina Müller, our statistician, and all this genetic epidemiology work is only possible in the context of collaboration with the Genetic Institute, Thomas Meitinger, and in our epidemiological work in the CORA study, but also embedded in large international collaborations. While the title is already very descriptive, it is important to consider why do we do genetic analysis in arrhythmias. It is towards an improved mechanistic understanding of the arrhythmia pathophysiology, and this is to facilitate then individualized medicine or biomarker-based stratification, group building of specific arrhythmia subgroups or subpopulations, and then leading to targeted therapeutic approaches in the future. This is just to remind you that our genomes, while the sheer number of the genes are not so different from the worm or the fruit fly, but it's the total amount of genomic information that really makes a marked difference. And despite a huge homology on the genomic level of all of us here, we are aware of the great variance that we see, and the common variants are distributed like this. So if we talk about a common genetic variance, it is important that this is embedded in a more broader genomic approach, where you will see examples from candidate gene-based and then hypothesis-free genome-wide association studies, now also moving to the rare variants, next-generation sequencing, but importantly linking this information then to expression studies in microRNA, and I will not talk about these three today. We have started to look at the genetic information on arrhythmias, but by starting at the ECG. The ECG, in a very famous editorial, has been acclaimed as the cardiologist's best friend, and it gives a lot of information, even in these very basic traits. And ECG signals from a genetic aspect are markers and quantitative markers of myocardial electrical properties. They also are markers or surrogate or endophenotypes of arrhythmias, and as these quantitative traits, they contain a considerable heritable component. And you can dissect the components of the ECG very simplified in the RR interval as a biomarker for pacemaker function and heart rate, the PR interval is a biomarker for atrial conduction, and it's also an endophenotype or a risk marker for atrial fibrillation, the QRS duration for ventricular conduction, and the QT interval for a biomarker for repolarization that has also been linked to cardiac death. All of these have by now been analyzed in large international consortia with genome-wide association studies. I give you the initial examples of this, identifying a wealth of common genetic variants or genomic loci that are associated with modulation of these quantitative traits. This is to demonstrate that yes, in fact, these ECG signatures have heritable components that range from 30 to 60 in some cases, and the hypothesis was whether genome-wide association studies will help to identify new pathways or new arrhythmia genes, so to say, and then potential biomarkers for cardiac arrhythmias. And I will give you the initial example that started out in 2006 with Dan Arking and Arne Pfeuffer, that were able to identify this still most prominent signal that modulates QT interval, the NOS1AP gene, and in the consecutive analysis. Now this signal had been identified with only 200 samples, and then the experiment was expanded to 15,000 individuals, and all these signals are identified. Still, this strongest signal in these Manhattan plots, where you see the chromosomal localization and the threshold for genome-wide significance, identifying relevant hits in these sort of Manhattan skyscrapers that were identified as modulators of the QT interval. And it was no surprise that genes that have already been involved in the rare arrhythmia syndromes, long QT syndrome, also showed up as modulators in the common variants that modulate the QT interval. But the strongest signal still was something new, and the role and the pathophysiology in the QT was not known. And this is a slide summarizes all the data that since 2006 have been reported on the role of nitric oxide synthase 1 adapter protein, or NOS1AP. And it was only the initial publication that looked at the function of NOS1AP when overexpressed in cardiac myocytes, resulting in action potential shortening as an indicator that yes, indeed, NOS1AP regulates myocardial repolarization under cellular preparation. All the other experiments and publications on NOS1AP are again association studies with different types of arrhythmias, ranging from sudden cardiac death in the general population to most recently to as NOS1AP as a risk marker for drug-induced long QT syndrome. So again, this theme prevails. It is more association studies than function and causation of these common variants. I give you one example where the function really helps to identify specific loci, such as in this NDRG4, a signal that was also identified in this genome-wide significance, is one that was closest to this genetic location. Looking closer, there is a number of genes in this area, and it was the work of David Milan to dissect this locus and identifying not NDRG4 but GINS3 in a follow-up in fine mapping experiment, identifying all the genes in this area, and then looking at these genes and knocking down the genes in a zebrafish embryo, and as a result, only the GINS3 knockdowns revealed a electrophysiological phenotype that goes with QT modulation. When optically mapped and APD determined, then the knockdown of GIN3 really results in a shortening of action potential. And this is an example that from the genome-wide association, you have to go into fine mapping analysis, you have to go into the genes, and then, by example, with a zebrafish assay, look into the function of this gene that has been identified as a common variant. But what about the pathophysiological insight from genomic approaches and common genetic variants in specific common arrhythmia phenotypes? And I give you three short examples. One is on drug-induced long QT syndrome. The other one is on sudden cardiac death in the context of acute MI. and the last one is on atrial fibrillation. This is a candidate gene-based association studies that was performed in 176 patients with drug-induced long QT syndrome and compared to population control that had no history of drug-induced arrhythmia. And this slide depicts again the genomic location of the 1,400 SNPs in 18 candidate genes, and it was only one signal that survived this study in the replication analysis, and this is a gene and a genetic variant that came as no surprise. KCND1B85N had previously also been associated and functionally studied in the context of QT prolongation. And you can see only briefly that with the co-expression of this variant to the KCNQ1 channel, the kinetics of the channel are altered, and in a modeling experiment, the wild type E85 or the mutant E85N show differences with IKR block, and then only with an additional challenge on repolarization. Here in the wild type with 100% IKR block, there's only minimal action potential prolongation, but in the mutant channel only with 50% IKR block. In this model, you see early after depolarization suggestive of the arrhythmogenic mechanism in this. A second example where we are searching from common genetic variants identified in genome-wide association studies to the function of novel genomic loci is an experiment done by the group of Arthur Wilde, the AGNES study, Arrhythmia Genetics in the Netherlands, where a very specific phenotype is being evaluated. That is patients that have VT or VF in the context of their first myocardial infarction compared to an age and sex matched population that have a first MI without the arrhythmia. In 500 cases and close to 500 controls, this genome-wide association study had been performed, and only one signal surpassed the genome-wide threshold for significance, and this CAR1 Coxsackie adenovirus receptor protein has been identified, and